Antibody-oligonucleotide conjugates are a platform for delivering oligonucleotide therapeutics to cell and tissue types that have generally not been accessible to oligonucleotide therapeutics. The promise of oligonucleotide therapeutics is that you can design active therapeutic agents based upon Watson and Crick and Franklin base pairing rules. So it's a completely rational design based upon the sequence of a disease-related mRNA or disease-related RNA in general. And so the issue that has been plaguing oligonucleotide therapeutics, if you can knock out any disease-related protein, why aren't we using more oligonucleotide therapeutics on a daily basis? That is, it's been difficult to deliver them. So cells have evolved over time to exclude foreign oligonucleotides. Look what happens when you have a foreign oligonucleotide that gets loose in the population. We're now speaking 3,000 miles away from one another, or 6,000 kilometers from one another, because a virus figured out how to get their oligonucleotide material, their genetic material, into cells. So that was a challenge that we had to overcome, and nature's overcome it by taking advantage of receptors on cell surfaces. So what AOCs do is they use a monoclonal antibody to bind to a cell surface receptor, and attached to that monoclonal antibody or conjugated to that monoclonal antibody is an oligonucleotide therapeutic. And in that way, we can essentially use cell surface receptors that will transport our oligonucleotide that's bound to an antibody that's bound to that receptor into the cells, and then it can be released in the cells. So that way we get around the barrier that cells have built for themselves to keep foreign oligonucleotide material or foreign genetic material out of them. So with AOCs, in particular for the AOCs in skeletal muscle diseases that we're here talking about today, the AOCs are built around an antibody to the transferrin receptor. So the transferrin receptor is a cell surface receptor that normally takes up iron. And what we're doing is we're attaching a protein-bound form of iron, and what we're doing is we're attaching a monoclonal antibody that will bind to that receptor. And when the cell believes, or the protein thinks it's taking up an iron-bound protein, it's actually taking up the monoclonal antibody and the oligonucleotide attached. So we're essentially using that as a transport mechanism. In the case of Duchenne muscular dystrophy, we're trying to deliver a oligonucleotide therapeutic that's going to allow that oligonucleotide therapeutic to skip over exons in particular exons in the dystrophin gene, DMD. And of course, Duchenne muscular dystrophy is a disease that's associated with out-of-frame mutations in the dystrophin gene. And if you can skip over those out-of-frame mutations, you can get an in-frame protein that's essentially functional. So much of the work that was presented at AAN with respect to Duchenne muscular dystrophy was a demonstration that using this technology, using a monoclonal antibody to the transferrin receptor, you can effectively deliver an oligonucleotide therapeutic in a mouse model of Duchenne muscular dystrophy. And the data that we presented there quite clearly demonstrate that we get functional changes, we get functional improvements, we get increases in the amount of the protein that's formed. We're skipping over those out-of-frame mutations to create an in-frame protein, or in this case, a premature stop codon. We're allowing the disease model in mice to actually produce a functional dystrophin protein and restore their movement and restore some of their strength.